we're trying to understand the 5% of patients admitted to an intensive care unit who are younger than 50 years and without any known underlying medical condition. The hypothesis is that these patients, or at least some of them, perhaps many, suffer from inborn errors of immunity, that is, from germline genetic variations that make them vulnerable to the SARS coronavirus 2. The approach is to try to enroll as many patients as possible in as many hospitals and institutions as possible, in as many countries as possible worldwide, because this is a small proportion of patients with severe disease. And we know from the study of other infectious diseases, if you take 10 or 100 patients with a given infectious disease, there may be as many as 10 or even 100 different genetic variations, sometimes in 10 or 100 different genes underlying that particular infection. So if, say, there is a patient in Manhattan mutated in gene A, it may be that there's a second patient in Tokyo and a third in Sydney. So the only way to detect a signal suggesting that these mutations may be responsible for severe COVID is to enroll patients in Manhattan, in Tokyo, and in Sydney. So we have hundreds of hospitals worldwide working with us. We enroll these patients, we sequence their genome, we select candidate genes, and then we test them. We test these candidate variations biochemically and immunologically. The answer is a prudent yes, because Helen Su and our lab identified inborn errors of immunity underlying severe pneumonia caused by two viruses, influenza virus and rhinovirus. Influenza virus is the virus that causes flu, influenza, and rhinovirus is one of the viruses causing the common cold. But there are patients with severe pneumonia caused by both viruses. So these genetic disorders only might be involved in some patients with severe COVID pneumonia. It depends. We don't have a crystal ball. What I can tell you is that for some infectious diseases that we studied in the past, such as microbial diseases in particular, the discovery of inborn errors led to efficient and novel preventive and therapeutic measures. I also want to stress that the implications can go beyond the genetic cases, because understanding the genetic basis of a particular infectious disease in a relatively small subset of patients with that particular infectious disease is also illuminating beyond the genetic cases patients who suffer from the very same infectious disease for other reasons. They can be treated. You're essentially providing the patient with the component that his or her organism is not producing in sufficient amounts.